Thank you, and welcome to this symposium. I think it will be quite interesting for me and hopefully as well for you. What I would like to talk about in the next few minutes is this new emerging paradigm of treating data as a resource. And then secondly, I'm going to talk about just a few of the tools and approaches, particularly focusing on some of the approaches for managing data these days that I think will probably be new to at least many of you. I'm going to tell two stories about some of the societal benefits of treating data as a resource that date back, in some cases, a few decades, up to more recent times. And then I want to talk about some of the milestones for biodiversity and ecology data that will help, I think, set the context for the remaining presentations that you will hear today. So first of all, a definition. This comes from a modification of Thomas Kuhn's earlier definition about paradigm. And it's basically the set of concepts and practices that define a scientific discipline at a particular period of time. And the paradigm shift looks like this. This has been the paradigm for many decades previously, in that, we come up with an idea, we design an experiment or a study, we do the field collection, data capture, do the analyses, and then end up with a publication. And then in many respects, the data disappear after that. They're either put onto a hard disk or a laptop or sent off to file drawers. And they're very oftentimes impossible to find several years after the study has been completed. So the new paradigm, or the emerging paradigm, is to treat data as a resource, as you see there, so that data can then be discovered by others. It can be integrated with potentially other data sets, maybe for a meta-analysis. It can result then in additional publications and new data that possibly, again, represent integrated data resources from not only that study, but from others as well. And then the circle can go on ad infinitum, as we see here. This data resource, again, can now be mined and used by others over time. There are a lot of reasons for this new change in paradigms. Some of them deal with the science questions that we're trying to address. Others deal with imperatives put forth by societies and publishers, also research sponsors, and then increasingly we're seeing interest on the part of the public with respect to making data available, making scientific studies more transparent and reproducible. So the first, again, major driver is the science that a lot of us in this room are attempting to address. Some of these grand environmental challenges, global climate change, changes in energy sources, things like infectious disease, a whole array of very complex topics that require data from many, many different sources, a lot of it historic data going way back in time, as well as data crossing many different disciplines and scientific domains. The societies and publishers have increasingly been drivers in this regard. Going back to 2011 was one of the, I think, major events where a number of societies and publishers got together and they created this joint data archiving policy that says that a journal will require that the data supporting the results in a paper should be archived in an appropriate public archive, and then explicitly recognizing that data are important products of the scientific enterprise. And this led to quite a number of follow-up activities. The following year, the Dryad Digital Repository was formed to accept data products associated with publications from many ecology, biodiversity, and evolution journals. This was followed up a little bit later with the PLOS, Public Library of Science statement that required all data underlying the findings in a manuscript be fully available without restriction. And then just this past 2 years ago, Nature adopted the tools for transparency and in ecology and evolution. And this checklist really focuses on several things, both empirical studies, meta-analyses, and then it provides a checklist for article referees and for editors of various journals to make sure that the publications provide not only the data, but other software, algorithms, and so on that make it possible to understand how the results came about. A precursor to this was conservation biology, which adopted these checklists and guidelines earlier. Other drivers were research sponsor requirements. Back in 2001, this was in the United States, of course, the National Science Foundation released their policy, which said that the data associated with a publication or research project needed to be available by other researchers. And we've seen this go internationally. The Wellcome Trust more recently announced their guidelines, which were not only data, but also the software and the algorithms that were associated with the project needed to be available as well. And then the last is the public trust. And this has become apparent with a lot of reports that you've seen in the newspapers about ClimateGate and other issues associated with questioning scientific findings. A couple studies here that I highlight. One, really at the top there, Bionides focused on a number of issues, including poor quality assurance, quality control techniques, lack of metadata, and so on, that made it virtually impossible to replicate studies. Secondly, the open science collaboration, as you can see on the right, found that most replication effects were smaller than the original results when people attempted to replicate experiments. So this created, of course, a lot of consternation on the part of researchers, societies, publishers, and others. More recently in the U.S., with respect to the National Climate Assessment, as you can see on the left side, this is a huge effort on the part of scientists in the U.S. to periodically assess the state of the climate and impacts of climate change. So this is a 3-year effort, over 300 experts involved, a very large advisory committee, a huge amount of work. And in this most recent release of the National Climate Assessment, significant effort in terms of many decades worth of man years or human years went into providing the associated website, which provided details about how every single result was obtained for the National Climate Assessment. So I think this was about, best I can tell, somewhere around 50 years worth of effort in terms of the number of people that were involved in going through and mining all of the details about where these different results and graphics and so on came from and making that available to the public. And then very recently, of course, we've seen the FAIR principles being released. This is through Force 11, a group of publishers, scientists, and others. And FAIR, if you're not familiar with the acronym, stands for Findable, Accessible, Interoperable, and Reusable. And many societies and publishers have adopted these guidelines, and they are now affecting policies that you see in journals and associated with professional societies and even research sponsors as well. So I didn't want to spend too much time with respect to tools and approaches, but I thought I would mention just a few of the tools and approaches that I find particularly useful. Here I'm showing the data lifecycle that was developed in the DataONE project that I'm associated with. So starting at the top, it goes from planning through collection, assurance, description or metadata, preservation, discovery, integration, and analysis. Clearly that does not necessarily need to be sequentially done, but some of the key tools that I would point out to you very briefly. DMP Online for planning is in the French language. It's available for you to use. It's an incredibly valuable free tool for helping to create data management plans. MySQL or MySQL and Specify has been adopted by a large number of museums for handling biodiversity collection data. It's a free package as well. R is, of course, universally used for QA/QC, and I include JMP in here. Even though it's a commercial product, I found it to be very useful for really complex quality control charts that are very difficult to program and using other means. In terms of description there, ecological metadata language and Morpho, which is a metadata management system that facilitates creation and management of ecological metadata. And then, of course, Darwin Core, which a lot of you are probably familiar with as well for handling specimen data. In terms of preservation, re3data.org, which enables you to easily discover data repositories that might be applicable. And then I mentioned OneShare and OneSearch as DataONE products, again, from the project that I'm involved in. We'll hear more about GBIF in one of the talks today by Donald Hoburn. For integration, again, MySQL, and then a number of tools for analysis, one of which, again, is a commercial product, MATLAB, but RStudio and R are used quite frequently for a whole variety of different types of analytical studies. There are a number of other, quote, tools or services that are commonly used these days. One is controlled vocabularies, and I just show one of the earlier ones here, but there are quite a large number of controlled vocabularies that help you provide context and descriptions for your research projects. This really facilitates integration and analysis by others down the road when these keywords are included in metadata descriptions. Similarly, we see a big push in terms of ontologies. This is started as a European initiative here, the environment ontology, and it's growing by leaps and bounds. It's becoming widely adopted by various communities to help better provide semantic context for their particular research projects. One tool that I want to introduce, this is a new one available through DataONE, and one of the questions that people have always had is, how do I show the provenance for where a particular result came from? So we've been working with some of the standardized approaches to develop what's called our provenance tracking system that allows you to look at a particular script, an R script, for example, or a particular graphic or a result, and then show the data that went into creating that particular product, and then any subsequent derived data products or analyses that came out of that. So on the left here, you can see this particular script uses data from these 4 different sources on the left, and the one I show here is alkane.csv. This is a hydrocarbon, impact of hydrocarbons on fauna in Alaska in this particular case. And then we see the script. We can actually look at the script itself in the middle, and on the right side we see the derived products, graphics, and so on that came from that. And we can look back and forth amongst the various precursors and products. And this has just been released, and we provide tools to help provide the annotations that support this using both MATLAB and R. And then there's a generic tool called Yes Workflow that can also be used as well. We have a lot more details about this on our website. So those are mostly the tools. I wanted to spend some time talking about the approaches, though, because I think this is really, excuse me, this really highlights how a number of groups have adopted more open approaches for managing, sharing, making their data available, and making it more reproducible and transparent. The first example I wanted to give is from the Ocean Health Index. And this is a project that started at the National Center for Ecological Analysis and Synthesis back in 2011. The first, I think, analysis came out in 2012. And here they're looking at the health of all the world's oceans according to a whole number of quantifiable metrics, as you can see here. In the center is the title of the paper that I want to highlight briefly, which is Our Path to Better Science in Less Time Using Open Data Science Tools. And in particular, they're focusing on those tools that I show on the right, R, Git, RStudio, and GitHub. And importantly, what this group did was they said, back in 2012, and I'll have to point to this, if you see in the lower left, there's a big circle with a large dark area and then a lighter area as well. So anyway, the bottom line is the size of the circle equates to the amount of time that it took to actually do the work that year. And then the dark shows the amount of time that was spent actually on science. And then the lighter gray shows the amount of time that was spent actually on the data or informatics side of the equation. So back in 2012, they focused in terms of developing their protocols and initial workflows and analytical procedures. Took a huge amount of time. A lot of it was focused on the science. And then after that, they said, this takes so much time and effort. Let's figure out a way to expedite this for future years. So the whole team got together and said, okay, let's adopt an open approach for doing this. So in 2013, that was their first year in terms of doing this. They spent less time overall, but a lot of the time was focused on the data and informatics part of it. So again, in 2013, you see a lot more of the light gray color. It got easier in subsequent years, as you can see by the size of the circle getting smaller and smaller. So in 2014, a lot less effort was involved in terms of replicating the Ocean Health Index measure for that year. But still, a lot of it was spent on the data and informatics side of it. So again, getting everything into R and RStudio, making the algorithms reusable by others, and so on. In 2015, it got even less time, and more of the effort was spent back on the science as opposed to the data side of it. And then in 2016 and 2017, again, a lot less time was spent, and a lot more of the focus in terms of energy and time has been spent on the science as opposed to the informatics. So they've shown that it gets easier and easier and easier over time, and they adopted this as a group effort. So they trained each other, they developed a toolbox to help make their life easier, and consequently it's been a huge success on their part in terms of making the work easier and also making it more transparent for others. Another approach for managing and sharing data was adopted by what's termed as Nutrient Network, or NUTNet is the short acronym for it. This started out of the University of Minnesota, and it's now gone global. And what this group did was they said, okay, we're going to develop this collaboration with standard approaches, standard data formats, and so on that will be adopted by all members of this network. And as you can see, with all the locations pins on the global map there, lots and lots of adoption globally for this. And again, it's benefited from everyone adopting the same standards and then the same approaches for sharing data, citing data, and so on. And this happened, I think, back in 2011. So this project has been around for quite some time. The next one is the emerging trend in terms of environmental or ecological observatories that are being formed internationally. One of the precursors was the National Ecological Observatory Network, which the initial design for that started back in 2007. And it's just really come online this year. So 2017, or past year, 2017 was sort of the startup date for this National Ecological Observatory Network. One of the main requirements is that all the data collected by NEON will be fully available and easily accessible by the broader community. So you can see all the locations where data are being collected. The NEON data portal is up there on the upper right, and you can see the types of data, and you can drill down further in terms of acquiring the data that you're interested in. And this is what it looks like as we go down further. You can apply a whole variety of different filters on the left to specify the data that you're particularly interested in. And on the right, you can see some of the data products. The first one there, wind speed and direction, air temperature above water on buoy, and so on and so forth, A to Z. And you can look at the timeline there, starting back from 2012 through 2018, the dark blue being that those data sets are available. So again, that fully came online in 2017, but some of the early sites were put in place much earlier. And you can download the data and the metadata and so on and use that for subsequent studies. The other one I will highlight is the project that I'm involved in, DataONE, which is a federation of data repositories. The idea here is that there are lots of environmental, biodiversity data repositories and others out there that host data that we might find useful in our research. But we may not be aware of all those individual repositories, and it really helps by having a federated, interoperable network to make it possible to discover the data that are held in any one of these different data repositories. So we can see the search site up on there, the search portal on the upper left, and then some of the data that are downloaded in the upper right there. And this is more or less what it looks like through an animation here. Let's say I'm searching for carbon cycling data. It will search the index of all the data products that are associated with all of the repositories that are part of this federation. And it will only pull out those data products that have anything to do with carbon cycling. Similarly, I could ask the same question with plant biomass, ocean nitrogen, and then avian distribution as well, and it will pluck out the data from those specific repositories that hold data that meet that particular thematic need. And similarly, there have been a number of biodiversity approaches as well. This is VertNet. It does fall on the heels of a number of other precursors. So there was a mammal network, a fish network, and ornis and ornithology data network as well, that were all based on the ability to go out there and harvest data from different collections using, at that time, the Z39.50 schema. And now this has recently been supplanted by VertNet, which sort of encompasses all of these different biodiversity vertebrate collections. And we'll hear about this, I think, also as part of Donald's talk about GBIF, which also includes VertNet products as part of the GBIF corpus of data. Okay, I wanted to spend the remainder of the talk focusing on two particular case studies, and I highlight here really one of my heroes, Rachel Carson on the left, and then one of my personal mentors on the right, Terry Yates. And I want to tell some stories that they were involved in terms of the research they did, which was really groundbreaking research. So Rachel Carson, some of you may know, she's a marine biologist. This is how she got her start, much like I did myself as a marine biologist way back in time. But she's responsible for this book, Silent Spring, that really made her name. She focused on looking at the impacts of insecticides on nature, in particular, a big focus on raptors and birds and other species. She's notable for quite a number of quotes. This is one that I find particularly compelling. Future generations are unlikely to condone our lack of prudent concern for the integrity of the natural world that supports all life. And one can argue that's still in some cases true today, that the attitudes that many people have. But what really underlay Silent Spring was a large number of existing data collections at the time. And this included not only citizen science observations, but also natural history collections that I'll show on the next slide, couple of slides here. So the observation that hawks and other raptors were declining in numbers came out of citizen science observations that are made in a place called Hawk Mountain, that you can see in the lower left. This is located on the Pennsylvania-New York border in the United States, and it had been a site of natural history observations of raptors since the 1930s. So there was a fairly extensive database that went way back in time. There were a couple of years during the war years of around 1944 that the observations were discontinued. But you can see that period when DDT was being used via that straight dark line there. And the upper graph shows the ratio of juveniles to adults. So that meant that during that time, there were almost no juveniles being produced, or a very small number of juveniles being produced when DDT was released into the environment. Similarly, we started to see a reduction in the number of eagles per hour in the lower graphic there as well. When DDT was outlawed, then after that you can see the upswing in terms of the production in terms of juveniles as well as adults. So this was explained further in a paper by myself and colleagues, including Keith Bildstein, who's the manager for Hawk Mountain Sanctuary on the left. Again, this was one of the really major citizen science efforts that goes back many, many decades and is used as a big success story for that whole movement. So what it ended up being related to, at least partially, we now have a lot better understanding of the impacts of DDT and other organic chlorine and other chemicals on species in the environment. But as you can see, you may not be able to see the dates, but if you can look towards the right end of the graph on the upper left, you can see that there was a precipitous decline in terms of eggshell thickness. And this was, in this case, this was for British peregrine falcons, but it was also proved to be the case for a large number of other raptors as well. So eggshell thickness created, reductions in eggshell thickness created lots of breakage in terms of eggs and led to that reduction in terms of numbers of raptors that were being produced. Lots of studies emerged from this, many of the really groundbreaking ones I highlight on the right side there. The second study comes from a little bit closer to home for me. I live in Santa Fe, New Mexico, which is just to the right of the X that you see on the map there. This part of the country, the Four Corners region, is notable for all of those grand stone mountain features that you see coming out of the southwestern U.S., Battleship Rock and lots of others that are sort of the iconic images of the Southwest. Back in 1993, we had an outbreak, and this was a sign that showed up in medical clinics, and let me read that to you in case you can't see it. But attention, if you have a fever along with muscle aches and pains, please stay in your car. Don't come into the clinic. We will come out there and treat you in your car, or attempt to. And This turned out to be hantavirus pulmonary syndrome, but it took a while to figure that out. And initially, in fact, a lot of the public and congressional hearings were held because they thought this might be biowarfare that was taking place. But it turned out to be a hantavirus that was associated with this little guy on the left here, Paramyscus maculatus, the deer mouse. And what it does is it causes initial flu-like symptoms, but then after about 3 days of incubation, very rapid onslaught of water and blood filling the lungs, and then pretty quickly death occurring. 70% mortality initially, and then now with treatment, the mortality rate is still about 38 to 40%. So it's still a very deadly virus. Again, within a month or two of when that outbreak occurred, the Centers for Disease Control came in and again identified this was a rodent-transmitted virus. And that led to lots and lots of questions. What causes this? Why is it? What conditions in the environment make this happen? Are we likely to see it again? And so on. And as it turns out, it is quite closely related to climate conditions. We have a cycle in our part of the world called El Nino, La Nina. And in this case, El Nino leads to increased winter precipitation, which really affects the spring plant productivity. So we have, after an El Nino year, we have very high plant productivity. And then the following year, we have a real upsurge in the number of deer mice and other rodents that are come into being, as you can see in the lower left here. So the graphic demonstrates that on the right side with, again, high years of precipitation are associated directly with high amounts of vegetation. And then the following year, so it's a one-year offset, then we have high production of Paramyscus species and other rodents. So Terry Yates and colleagues went back and looked at and figured out that this cycle was what was responsible, but then they asked a lot of other questions. Can we relate this to other high precipitation events in the Southwest? The answer was yes. And they did this via not only the collections and the data records for numbers of mice being captured at different sites in the Southwest, but then also through preserved tissue samples and blood samples from mice that had been collected and stored at the University of New Mexico. This then led to the ability to predict when we might expect outbreaks of hantavirus pulmonary syndrome, which you can see on the left. This was another landmark paper. And then on the right, though, you can still see that we still have continuing outbreaks of that. And then the black line is the mortality rate. So even with identification and forecasting, we still have about a 38%, 40% mortality rate from this particular viral disease. But then Terry asked the question and said, okay, knowing what we know about evolution and biogeography and so on, we know that rodents came into North America via the Asian land bridge back in 20 million years ago. And then over time, they migrated into South America about 7 to 9 million years ago. They moved past the Isthmus of Panama. So the question is, shouldn't there be hantaviruses throughout both North and South America? And that led to a whole series of studies by Yates and others. And all of the ones that are highlighted in yellow here are ones that are associated with human mortality. So some of them just carry a hantavirus that only affects or is carried by the rodents, but many of the others have been responsible for a huge number of deaths and illnesses throughout both North America and South America. So again, the ability to discern this was based upon the fact that we had lots of understanding and lots of data associated with mice counts, productivity over the years, as well as the ability to go back and look at specific specimens from mice that were trapped and then relating that to the presence of hantavirus, and then also looking at epidemiological records as well to look at the impacts on human populations. So in the last couple minutes here, I just basically wanted to set the stage a little bit for some of the future talks that we'll hear today. And I wanted to step back in time a little bit and look at some of what I think were some of the landmark events. I'm sure I'm missing quite a few. I would love your feedback, and as well as my colleagues up here, in terms of any landmark events you think I'm missing. But I want to step back and look at the top. Usually I'll have a science activity at the top here, and then I'll get into some of the informatics and other activities on the lower part of the top bar. And then below the red arrows, I'll show some of the key policies that I think have had a big impact. So back in the 60s through the 1980, I think two big events there. One was the International Biological Program. This was a massively endowed program to really start looking at ecosystems worldwide. There was many countries involved, and one of the results of that was that there are simply no data available now, or very little data are available from this entire effort, but there were lots of publications that came out of it. And this was due to the fact that those that were involved just simply could never agree on data sharing policies and never adopted those across and even within individual countries. Another landmark event, I think, was the formation of CODATA, which we'll hear more about in one of the future talks from Simon, sitting on the front row here. Moving up 2 decades from the 1980s to 2000, a couple of landmark events there. I think one was the formation of the Long-Term Ecological Research Network in the U.S. You can go along the top here, the International Geosphere Biosphere Program, a fairly lengthy international program, many millions of dollars and euros equivalent spent on that. But as you can see in the lower left here, I show that there are only really two major legacies from that entire effort that are still remaining today. One is global soils and global land cover. Much of the other data, again, has been lost or is inaccessible for one reason or another. We have the International LTR program in 1993. NCES, the first major ecological analysis and synthesis center in 95. A number of informatics milestones there, TADWIG, Z39.50 protocol, Federal Geographic Data Committee that created the geospatial metadata standards, Specify for taxonomic data collections, and then Earth Science Information Partnership. And then the first real policy that I could come across was the LTR site data policy for how data should be managed within a site. And now we move up to more recent times and we see some changes. We see lots of new infrastructure efforts, Nutrient Network in the very top center, Ocean Observatory Initiative and NEON, many informatics associated efforts here, GBIF, K&B, one of the first public domain data repositories, the ISO 19115 metadata standard, ecological metadata language, GeoBond, DataONE, VertNet, EarthCube, Dryad, a public repository I mentioned previously, Copious, UDAT, LifeWatch, and followed by ERIC, the European LTR, and then EMVRI and Globus B. And then on the bottom, we had a couple policies. The one I mentioned previously was the NSF public policy in 2001. LTR started tightening up the requirements for data sharing in 2005. And then we have a whole array of policies that started proliferating starting in about 2011 up until today. And we will hear more about a couple of these efforts. Don will be talking about GBIF. Dirk focusing on Globus B and the work with essential biodiversity variables, and then I think both of them may possibly touch upon GeoBond as well. And I'll conclude here with just some big thanks to lots of colleagues that helped out with various aspects of this presentation. So thank you for your attention, and I think we just go straight into the next speaker.